I'm Ross Camage. I'm a clinician. I'm a medical oncologist, and I run the lung cancer program. And you know, it's funny because one of the things is, and what I think really emphasizes the strength of a cancer center is when there is a dialogue between the laboratory and the clinic. And so I would like for my co-chair to introduce himself.
Yeah. Hi, Ross. So I'm James DeGregory. I'm one of the two deputy directors of the Cancer Center. And I also run a basic research lab that focuses on actually two cancers. We focus on both lung cancers as well as hematopoietic malignancies like AML. And, you know, we have intimate collaborations with a number of clinicians. So I call myself a basic researcher because I don't work in the clinic. I have a PhD and not an MD. But I'm continuously collaborating with my clinical colleagues, and really it's the power of all of us working together that makes the research really move forward.
All right, next slide, Colin. So I'm going to illustrate a little bit of that, and I know Ellen's on here, but this is one of my patients. This is Emily, and together with her kids, Paige and Brady, and I want to illustrate some of the things that we've been able to figure out through that clinical and laboratory collaboration. So Emily, who, just to be clear, has never smoked, developed advanced lung cancer, actually, while she was pregnant with Paige. And Paige is a baby. I'm sorry, while she was pregnant with Brady. And she's really done fantastically well. Initially, we found that she had a specific abnormality in her cancer we were able to treat, but then her cancer very quickly became resistant. And so look at that little picture I've got there. So there's a little wiring diagram, a battery, some wires, and a light that goes on. And what we do with a lot of our therapy is we say the instructions that are coming from the mutation in somebody's cancer are like the battery. They're shouting the orders to turn the cancer on, the light. And so many of our targeted therapies, what they do is they have that little green switch. They just sort of stop the signaling and the light goes off and the cancer goes back to sleep. And then this is an animation, so we'll see how well this works. Colin, you have to going to be light on your fingers here. Next slide, please. So it should be an animation. So when a cancer becomes resistant, there are two well-recognized ways in which that happens. So the first is the cancer just alters the same battery. So it gets additional changes in the same gene that say your drug doesn't bind anymore, I close down the same switch, and the same loop turns back on. Next slide, Colin. But we looked in Emily, and that wasn't the mechanism in her. So the other way that's well recognized is you can sometimes see that other genes become mutated, other things that can drive other cancers, and they can come in as a bypass loop. So that switch is still up. But if you bring in another wire that goes through a different pathway, it's somebody's cancer. And we looked for everything that was known in Emily, and no, we couldn't find what was doing that either. Next slide. So what we did was we said, we're at the cutting edge of human knowledge. What we actually did was we took a biopsy of Emily's cancer and we were able to grow it in the lab. And in the lab, we were able to find that, yes, indeed, there was another pathway turned on, but through a means that we still don't know why. But we were able to, having identified it from actually looking at the growing cells, to figure out what treatment to give Emily. And we added in another pill. She's just on a pill. When she goes running, I don't know, Ellen can tell us, but sort of, multiple times a week. And she's now had a complete response, no evidence of cancer for nearly 3 years now. So there, the challenge is, now that Emily's cells are growing in the lab, can somebody like James figure out why? And that, once we figure that out, it's not just about helping Emily, it's about figuring out what's going on. And now can we develop a test that doesn't involve growing the cells in the lab, but actually you could do on a normal biopsy to send it out around the world. So that is where we are using funds. Next slide. James, you're up.
Yeah, so just to give a little background on what my own lab does is that, we're trying to get better understanding of the causes of lung cancer and their vulnerability. So really working from, both a cause and a treatment perspective. So from the cause perspective, obviously the ideal I mean, you know, obviously we know why a lot of lung cancers are caused, and that's because people smoke cigarettes. But there's still, depending on the country, 15 to 30% of lung cancers that are in never smokers. People have never even touched a cigarette. And so from the perspective of my lab, we're trying to understand how the fact that we get old contributes to that. So, you know, a lot of people always try to ascribe sort of an extrinsic cause for every cancer. But for a good chunk of lung cancers, as well as some other cancers like AML, there doesn't appear to be anything that we could necessarily do to avoid it. It's just the fact that we get old. And as we all know, you know, we can mitigate our aging to some extent by doing everything our mothers told us to do, like not smoking, eating a good diet, and exercising, but we are going to all age. And so we're trying to understand how those connect. And the better understanding of that should then lead to ways to hopefully prevent these cancers. And in addition, we are developing interventions to both prevent and treat these cancers. So we have, for example, figured out that we can prevent some of these aging-associated lung cancers, at least in mice in the lab. That doesn't mean this is necessarily going to translate into humans, but we are involved in some studies with our colleagues at the Veterans Administration, where they are testing some interventions in people, mostly smokers and former smokers, but they're basically testing whether if they give them a very simple and non-toxic treatment, if it reduces their chance of progressing to lung cancer. And then we, of course, then go back and take any information we learn from that and then use laboratory models where we can really do more cause and effect type experiments. So that's some of the stuff going on in my own lab, but I should mention that all of this work is really team efforts. So it's not just my lab, but my lab plus the clinicians, plus people who do bioinformatics. You just heard from Dr. Schulich and Riley that, you know, understanding a lot of this complex data sets really requires people who can crunch the numbers and you know, put two and two together, put it in a much larger way.
Yeah, I think on that point, I just wanted to reflect back that Dr. de Graury here is really representing all of the basic scientists that have had some impact on this and continue to work on this. And I'm really representing all of the clinicians and clinical researchers who work on lung cancer. So we're the people you can ask questions for, but we are by no means representing, you know, ourselves. Indeed, one of the things I've always taken a pride in is, you know, shouting out about the team. So this slide, they asked us to say, well, you know, give us a few bullet points as to what you think the future is going to hold. So I'll give my 5 cents here. So, you know, Emily, who I gave you the example, is a great example of extending out benefit as we turn what seemed like one disease, lung cancer, into multiple different diseases on a molecular level. I now have multiple 10, 15-year survivors. And 20 years ago, the chance of surviving 1 year if you had advanced lung cancer was about 25%. And now I'm having conversations with people about, can they become pregnant? Can they get married? Can they get promoted in their job? I mean, it's just incredible. The other thing is understanding that even when we think we've kind of split this into uniform lumps of people, some people's cancers haven't read the textbook. Emily would be a great example of that. She was supposed to do great on the initial targeted therapy. From several years of our work, she was expected to have several years before we had to do anything, but we were scrambling within months. So why is that variation even within our homogeneous groupings? There is variation. We're starting to understand it, and how do we act on it? And then if you really want the blue sky idea, great, we have treatments that can get your cancer to essentially disappear when you're on the treatment. But if I stop the treatment, the cancer will come back. It's not a cure. So how do we get 99% depopulation of the cancer? Amazing results, and yet not cure it. And that idea of just getting it over the finishing line, finding that little niche of persistent cells, which are not actively growing, they're just surviving, that's certainly something I'm passionate about. And then let's not forget that the best way to treat cancer is not to get it in the first place, or to get it when someone can cure it just with surgery or radiation. And can we use all of our fancy understandings about diversity in cancer and turning it into a personalized detection methodology? And that is also going on.
I would just add to that. I think that, you know, that first bullet point is so critical, extending out benefit and extending out benefit with therapies that are tolerable. So, you know, in the old days, shall we say, you might live a year, and that year was spent, you know, under the effects of chemotherapy and radiation therapy. So it wasn't necessarily a fun year either. But, you know, just to give a personal story, 3.5 years ago, I got an email from my aunt, who was actually, I have 6 aunts, and I have to say she was my favorite aunt. She still is my favorite aunt because she's still around, that she had metastatic stage 4 lung cancer. And so the first thing I did is I emailed Ross and Bob Doble, and they told me to do exactly what I already thought I should do, which was, you know, force her to go to MD Anderson. And of course, she was, you know, in Austin, Texas. She had her own personal doc. And she's like, I really want to stick with my doc. And I go, no, you got to get that second opinion at MD Anderson. And so she did end up going through, actually, she didn't have to leave Austin, Texas. She got to get someone in the MD Anderson network, and she got on immune therapy, and she's still with us and doing great and been going on cruises and enjoying life. In fact, the one thing she hated about the pandemic is because she couldn't keep going on her cruises. So it's a great example where someone who would have had a horrific prognosis, you know, just a couple of years before she got that diagnosis, has, you know, is now 82 and still enjoying life. And, you know, we don't know if it's going to be a forever cure. I kind of doubt it, but, you know, she's enjoying life and on a very tolerable therapy that wasn't imaginable even 10 years ago.
That's fantastic. Well, I think the next one might be our last slide. Keep me right. Next slide, Colin. There we go.
Right. So I guess I'll start off with this one. And this is really important, you know, so I have an endowed chair, and, you know, that provides money that, you know, that we haven't exactly defined what we're going to do with in a grant. So when you get a traditional grant, you have, you know, your aims, and you lay out what you're going to do. And you even sometimes have to have milestones and so forth, which is all good because people need accountability for their money. But the beauty of some of this philanthropic money is it allows us to think outside the box and really initiate new projects. And I'd say at least 2/3 of what is going on in my lab right now was initiated with basically that sort of money, sort of unrestricted funds that now has received much bigger funding because it actually worked. Now, that's not to say that all of these crazy ideas work. A lot of them do end up in the trash heap. But, you know, being able to take some risk and do something that you really wouldn't be able to attain traditional funding for is just so critical. And then the stuff that then works, you can then go for the bigger money for.
Yeah, and so, I mean, for those of you who haven't met me before, my accent is British, and, you know, I grew up in a place where, you know, the government sort of funded anything, sort of funded everything, although not very much, and philanthropy wasn't really part of the DNA of the culture. I moved here, and I think I really, you know, the sort of scales fell from my eyes, and I could really see what philanthropy could do. I mean, the grant process, I think, is important. It certainly took a bit of a pinch over the last few years. It's, you know, it's politically controlled to some extent. But you know, you have to write a grant, you know, that might take 100 hours. It might take many months to be reviewed. It might take many more months before the funding actually arrives. And then the NIH decides they're going to pay you, you know, 89 cents on the dollar or whatever. With philanthropy, James and I can come up with an idea today and we can start it tomorrow. So it really is kind of, you know, lights, camera, action, and you can get going on it. And then the other thing, and this really shouldn't be underrepresented. You know, I train many very talented clinician scientists, and I say to them, if you're good, you will get job offers. Many of them often get terribly threatened when they suddenly start being recruited, and that's exactly what happens. It's very competitive in academia, and people will try and, you know, steal the best people. By giving somebody, so James and I both have endowed chairs, that essentially, you know, sets bedrock in our feet, and we stay here. And the person who gave me my endowed chair was so that people wouldn't have to go and travel to other places. And I'm incredibly grateful to her because it's certainly given myself and my family stability and allowed me to bring what talents I have to Colorado. So philanthropy makes a huge difference.